many of you have noted. What I'm going to do is I'm going to skip the introduction with the sideboards. You can write it down, but it's all will be there, and then we'll cover it throughout the lecture. But I'll let you write this down just so we can catch up a little bit on time. And where we're going to begin is we're going to begin with the discussion where we left off last time, was the concept of anchoring junctions. And the anchoring junctions come as two types. They come as the hemidesmosomes and the desmosomes. When you see the term hemidesmosome, think attachment of cells to the basal lamina. When you see the term desmosome, I think attachment of cells to one another. And you remember from yesterday, or day before yesterday, that we talked about the characteristic of an epithelium always sitting on an extracellular protein matrix called a basal lamina. So this is the basal lamina. And that provided a certain orientation to these cells such that there was now a basilar surface and an apical surface. And where we left off was to describe the attachment of the cells to the basal lamina through a structure called a hemidesmosome. So I'm going to enlarge this and describe the hemidesmosome. So this box is meant to be an individual epithelial cell. Then sitting on the cytoplasmic side of the basilar membrane down here is a group of proteins, which we're going to basically call a protein plaque. There's some 30 different proteins in here, and if you get into a cell biology course, you'll actually get down into the weeds on this and talk about all the different proteins that are in the protein plaque. Some of them are regulatory, some of them are anchoring proteins, but we're just going to say there's a protein plaque. Then that protein plaque contains sites where these intracellular cytoskeletal proteins attach. Now, the intracellular cytoskeletal proteins could be actin, or in cells we'll talk about a little bit later today, called keratinocytes, it could be keratin. You may remember that keratin is an intermediate filament protein. But on the cytoplasmic side of the protein plaque, we now plug in these cytoskeletal elements, either actin or keratin. But remember out here, there's an extracellular matrix, which is the basal lamina. And what we need is some mechanism to anchor this structure of the plaque and the cytoskeletal proteins to the underlying basal lamina. And what we do there is now have a transmembrane protein that's locked into receptor sites in the protein plaque, but extends across the phospholipid bilayer and goes into the extracellular space. We have a whole series of these. These proteins whose feet are tied to the protein plaque, whose waist is in the phospholipid bilayer, and you can imagine their hands being out in the extracellular matrix. These proteins have a name. These proteins are called integrins. And the function of the integrin is to link the protein plaque with the extracellular basal lamina. The protein that you most commonly find out here that's binding the integrin to the extracellular matrix is a molecule called integrin. I'm sorry. It's a molecule called laminin. So the laminin is a molecule outside the cell. It's embedded among the proteins in the basal lamina. The extracellular portion of the integrin molecule binds to that molecule laminin. That ties it into the protein matrix. But the protein is a transmembrane protein. Integrin is a transmembrane protein so that its feet are in the protein plaque. So now with its feet in the protein plaque, anchored in the protein plaque, and the protein plaque then is in turn anchored to the cytoskeleton. This makes a relatively strong bond, allowing the epithelial cell to be anchored to the underlying basal lamina. So it is classically an anchoring junction, holding this cell down to the basal lamina. To give you some sense of how important these proteins are, there are some individuals who have mutations in the integrin molecule. The integrin molecule doesn't assemble properly, doesn't come across the membrane properly, doesn't make a proper fit with the extracellular protein matrix. These individuals have a very unpleasant life. The least little bit of friction, the least little friction between the epithelium and the underlying basal lamina, we'll see a little later being the epidermis and the dermis, that kind of friction will tear these cells loose and basically tear the epidermis loose from the underlying basal lamina. And they end up with blisters all the time. It's a horrible disease. But this is just to point out to you how important it is for these transmembrane proteins, the integrins, to lash the cell down to the extracellular matrix, the basal lamina, to hold it fast to the basal lamina. So hemidesmosomes are holding the cells fast to the underlying basal lamina, and over here is their cousin. The cousin to the hemidesmosome, I need to spell out, right, the hemidesmosome, the cousin to the hemidesmosome is the desmosome. And again, the desmosomes are forming attachments between cells. So I'll lay out an example of 3 cells here. And initially, this structure looks pretty similar. Here on the cytoplasmic side of the membrane sits a protein plaque, probably a different cohort of proteins than we see in the hemidesmosome, but some of them must be the same because the protein plaque will be linked into the cytoskeleton. This could be actin or keratin of the cytoskeleton. So again, the protein plaques are connected to the cytoplasm through the cytoskeleton. But they now also have to have a transmembrane protein. And the transmembrane protein here is one that goes across the phospholipid bilayer again and out into the extracellular space. But these structures are symmetrical. These molecules are called cadherins. And we can describe the cadherin as being an extracellular me try that again. We can describe the cadherin as being a transmembrane. A transmembrane protein that binds to cadherins in the extracellular space. So one of the ideas that comes out of this is that although epithelial cells sit on a protein matrix called the basal lamina, that protein matrix does not extend up between the cells. So in order to hold the cells one to another, there have to be these transmembrane proteins that link the two cells together. And so you can imagine, and I'll try to do it this way, so that if my right hand is meant to be one of these green cadherins, my elbow would be stuck in the protein matrix. Then my forearm, my forearm would go across the phospholipid bilayer and extend into the extracellular world. But then on the other side, in the other neighboring cell, there would be the same arrangement, with my elbow stuck in the protein plaque, my forearm going across the phospholipid bilayer, and my hand out in the extracellular space. And then my hands are now going to link one to another to hold these two cells together. So now the cadherins are linking cadherin to cadherin in the extracellular space to link these cells together. Now, I've shown these two cells to link together. These cells would also be linked together. But this doesn't do justice to the system because, remember, this is a great sheet of cells. And all of these cells are surrounded by other cells. And each cell is linked to its neighbors, all of its neighbors, by these desmosomes. And in some cases, they're referred to as belt desmosomes because they go all the way around the cell, linking one cell to the next. So all these intercellular linkages, so these are anchoring junctions that link cells one to the next. So neighboring cells are linked by these anchoring junctions called desmosomes. You have a question?
Is the hemidesmosome / desmosome the complex of the protein plaque and the integrin / cadherin?
So the question here is one that I need to be careful about. So this entire structure, the integrins, the protein plaque, and the cytoskeletal filaments, that's all considered to be a hemidesmosome. You could probably even include the laminin in the hemidesmosome. That's all one anchoring unit, one anchoring junction. Then again, over here, this entire structure, the protein plaque, the cadherins going out into the extracellular space, and the cytoskeletal proteins in both adjacent cells, that's a desmosome. Now, it's worth taking a moment here to speak of the language. This is a hemidesmosome. It's a half. Now, it has different molecular components, but when it was first described, just from electron microscopy, when they first described these in the 50s and 60s, this was simply a hemidesmosome. It looked like a half of one of these. So a desmosome includes both the protein plaque and the cytoskeleton and the cadherins from this cell along with this cell. The desmosome is said to be symmetric. Yes?
Kind of discussed, but on the side of the molecules here, are those also plaques?
So this purple structure, I'm blithely calling a protein plaque. Now the protein cohort, the complement of proteins in this protein plaque is probably different from the complement in this protein plaque. And it's not just simply one or two proteins, it's a whole group of about 30 proteins. And they're probably important in regulating cell-cell adhesion. They're probably important in regulating adhesion to the basal lamina. They're also important in anchoring, in this case, the cadherins. They're important in anchoring the integrins, and they're important in anchoring the cytoskeleton. So they do all of those things, and it's a whole bevy of proteins. Yes? So the question here is, how close are these adjacent cells? These cells are remarkably close because the only space between them are the cadherin molecules. And so I'm not sure what the space is, but I suspect it's even less than the gap junctions. And the gap junctions are 20 nanometers. So any more? Yes?
So do the desmosomes not anchor to the basal lamina then? Or do they not come in contact with the basal lamina?
So the question here is, do these desmosomes come in contact with the basal lamina? And the answer is no. That the cadherins link cadherin to cadherin, whereas the integrins link cadherin to laminin. And as far as I know, there are no cadherins that link to the laminin. They always link to other cadherins, making a symmetric junction. Yes?
So would there still be a basal lamina and the other ones linking to the lamina?
So there's also a basal lamina down here. And there's also going to be a hemidesmosome. But now, instead of using cadherins, we're using integrins. Okay. Yes, in the back.
So if we get this right, actin or keratin is kind of an internal anchor, like their long chains kind of stretch into the cell to hold on to stuff to anchor to the next one, correct? So actin, plaque, you have your cadherin, they anchor to each other, and then back the other direction. But the actin and keratin are kind of internal anchors in the cell.
The actin and keratin are internal anchors within the cell. They are part of the cytoskeleton. In some ways, you can think about this as being a plant, where this is the outside of the leaves, and these are the roots down in the cytoplasm. Better move on. So the next type of junction I want to spend a moment on is the so-called tight junction. The tight junction is a type of occluding junction. And the function of the occluding junction is to block spaces between adjacent epithelial cells. So again, if we imagine this short series of epithelial cells, the orientation is such as they sit on a basal lamina. I won't draw in the hemidesmosomes. The hemidesmosomes will be linking these cells to the basal lamina. The point here is that the apical surface faces the lumen of a tube. And imagine now this lumen of this particular tube happens to be the small intestine, or even the stomach. So that the contents of the stomach or the small intestine could be lethal if it leaks down in between these cells. So these cells now are zippered together. They're zippered together at sites called tight junctions. Tight junctions are composed mostly of the proteins claudins, CLAU, claudins, and occludins. Now these are simple transmembrane proteins that are interacting one with the other in the extracellular space. So you can imagine them zippering the cell together, but they're not a very stout junction. They're not very strong because they're not anchored into the cytoskeleton like the desmosomes or the hemidesmosomes. So these junctions are easily torn apart, but their main function is to form a blockade so that molecules from the lumen of the tube are blocked from coming down between the cells. Now I tried to do this in the first period, I failed, so I'll try it again. what you're looking at here is two plastic bags, two Ziploc bags. And they're turned inside out. And sometimes they make this work and sometimes they don't. And if you take these two and you imagine getting these zippers lined up just right, which I'm failing to do, that you can now zipper these two cells, these two Ziploc bags together, one to the next. And when you zipper them together, you effectively now have the same thing as I've shown here. And so now, of course, if this were a proper cell, the top would be closed. And the tight junctions would hold much better. But now you can see how you have blocked, using the zippers of the Ziploc bags, you have blocked the space between these two cells so that things can't get down between them. So that's basically the function of these occluding or tight junctions. And the reason they're named tight junctions is, again, in the early days of the morphology, these two membranes were basically seen to be plastered one right next to another. The two membranes were tightly opposed. So that's how they got their name, tight junctions. Yeah.
So in between the zippers is where the desmosome is?
Okay, so then where are the desmosomes? The desmosomes are often found between the zippers. We'll also find sometimes gap junctions between them, but let's not get into those weeds. Anything else? Yes? So the terms tight junction and occluding junctions are interchangeable. And I'm just trying to introduce the three major types of junctions as being anchoring or occluding or communicating based on their function. But they have, before their function was known and before the protein complements were known, they were simply called desmosomes, hemidesmosomes for the anchoring, and then they were called tight junctions for the occluding junctions, and we'll see here in a moment the gap junctions or the communicating junctions. But they have these different functions, but they have old-fashioned names based simply on their morphology. Yes?
So if these are so easy to break, but they seem like really stomach acid, what happens when they do break?
Well, what happens when they do break? The fact is that you have many of these seams. It's not like the Ziploc bag with one. They'll have many of these repeated. But you're right in that as cells divide and replace, they've got to, new cells have to move into these sites and replace those junctions. And how that gets done is not clear to me, but that has to happen. Well then, let's move on to the next type of junction. Let me leave those up there and let me change this here. The final kind of junction I want to tell you about has the generic name, the generic term of a communicating junction. And the communicating junction, that term, is descriptive. It tells us that it allows molecules to move from one cell to the next. It tells us that we have communication between cells. So the term communicating junction refers to intercellular communication. Through channels. We're going to give these channels a name. They're going to be called connexons. And we're going to see each connexon contains 6 connexins. So again, here are these two neighboring cells. And there are sites where these cells come very close together, but they don't come as close together as in a tight junction. They leave behind a gap. And this gap is typically about 20 nanometers. Because there was a gap between these adjacent cells at these junctions, these got the name of gap junctions. So the microscopist, the electron microscopist, described these junctions as gap junctions because there was space between them. But then they began to do additional experiments, and they found that there were protein bridges between the cells. And then they started to do some experiments where they would inject molecules into this cell. And then they would see that molecules injected from this cell could now flow from one cell to the next. And by carefully selecting the molecules they were injecting into one cell and seeing how they were moving from cell to cell, they found that molecules with a molecular weight of less than 1,000 move through gap junctions. They then began to realize that these protein bridges were in fact channels. And that, so the idea that they had the protein bridges, then followed by the idea that you could inject dyes and have molecules move from one cell to the next, indicated that these protein bridges are a kind of channel. They are a kind of intercellular. They're a kind of intercellular channel. Now, if we were to take this view and imagine if we stick our head inside the board, or we rotate this cell 90 degrees, so that we're now looking down on this surface. So this is the surface of the gap junction. I should say the gap junction membrane. Now, one of the difficult things to recognize is if this is the gap junction membrane of one cell, there's another one out here. So this outside toward the plane of the audience, there's another cell. But then when we look at this gap junction, we see these clusters of proteins. We see that there are these 6 proteins arranged in a circle. Each of these proteins, each protein, each transmembrane, each transmembrane protein is known as a connexin. The 6 connexins form a connexon. And a typical gap junction may have several hundred of these connexons. So in this view, this side-on view, in this cross-section view, you now have these open channels between cells. But those open channels are in fact transmembrane proteins organized around a central hydrophilic pathway. You can read channel. Well, let's take a look at some of these in some slides, and maybe we can bring some simplification to this. Now, this first image, this is a really highly stylized view of the anchoring junctions. But I think it's worth looking at this stylized view because it simplifies things down to something that you can put your head around. So this yellow thing down here is the basal lamina. So that's the extracellular series of proteins. This green arrow structure is meant to be the integrin. So the integrin binds to the proteins outside the cell in the basal lamina. But it has its feet, if you will, or the base of the arrow here, is now attached to this castle-like structure, which is meant to be the protein plaque. So the protein plaque now can bind both the integrin, but also can bind cytoskeletal proteins. So again, this castle-like structure is really a cohort of about 30 different proteins. They're doing a whole bunch of different things, some of which is regulating the attachment of these cells to the basal lamina and may in fact have important implications for cancer and metastasis. But nonetheless, for our purposes here, we have the integrin to the basal lamina, the protein plaque to the cytoskeleton. That's a hemidesmosome. There's a similar structure up here called the desmosome. The desmosome, again, has this blue structure, which we showed as a purple protein plaque. On the cytoplasmic side of the membrane, it attaches. But out in the cytoplasm, this protein plaque attaches to the cytoskeleton. But simultaneously, the protein plaque will attach to this potato masher kind of structure, which is called a cadherin. The potato masher kind of structure is a transmembrane protein. Its feet are in this castle, but its head is out here in the outside world. In the outside world, these cadherins are sticky, so they stick one to another. So now these two cells are linked to each other by the sticky heads of the cadherins. That's how we hold the cells together. So they're anchored side by side through these transmembrane proteins called cadherins, but they're anchored to the underlying basal lamina by these structures called the integrins. Now, let me walk you through this because this is a fairly complicated figure. What it's first trying to show is how the phospholipid bilayers of two adjacent cells will be stitched together by the tight junctions. And that will bring these two membranes right one next to another and stitch them together with these little ovals, these little beads, which are representing the claudins and occludens of the tight junction. Now, to walk you through the structure that they're showing here, this is the cytoplasmic leaflet of the phospholipid bilayer. This is the extracellular leaflet of this cell. So we're going to call this the cytoplasm of cell 1. So we go across the two leaflets. We're now in the extracellular space. So this blue area is extracellular space. This is now the extracellular leaflet of the phospholipid bilayer of cell 2, the cytoplasmic leaflet, and now the cytoplasm. And what they're trying to depict here is how we use both phospholipid bilayers and we stitch these cells together using these extracellular proteins called the occludens and claudins. And fundamentally, by zipping the cells together, we now provide an occluded or blocked pathway between these adjacent cells. That's basically what these tight junctions are doing. And again, they're showing here how there are multiple tight junctions between these two cells. This is the structure I just tried to explain on the board. I think this is better explained here. But again, it takes a little bit of explanation because you have to describe these two red plates. The two red plates are representing the two phospholipid bilayers of this cell. These two red plates are representing the two phospholipid bilayers of this cell. These green flower-like structures are meant to represent the connexons. And if you count these individual units, there are 6 connexins forming one connexon, and the connexon is a channel. And what we're showing here is how those channels are linked in the extracellular space, sort of head-to-head, so that we now form an aqueous channel from one cell to the next. These are big channels. They're so large that they allow molecules upwards of 1,000 molecular weight to pass from one cell to the next. So the way they're formed now, this structure is going to be a gap junction composed of several hundred, perhaps, connexons, and each connexon is composed of 6 connexins. That's how they're organized. But these are proper communicating junctions, allowing communication from one cell to the next through the phospholipid bilayers, through those channels. So that's pretty much the story of the junctions. So what I want to do now is I want to now leave the story of the junctions and begin to bring together two tissue types into an organ. Now, conceptually, this is an important idea. When we describe an organ, what we mean is multiple tissue types working together. The simplest of all of the organs is going to be the skin. And the reason the skin is the simplest of all the organs is that it's an organ composed of 2 tissue types. The 2 tissue types we're going to describe here in a moment in skin are an epithelium and a connective tissue proper layer. And we'll spell some of this out before we do the drawings. So organs are composed of more than one tissue type. Skin is an organ composed of an epithelium called an epidermis on a connective tissue called the dermis. And you may remember we introduced tissues, we said that the dermis is connective tissue proper. Now, it's true that the epidermis is a stratified, squamous epithelium. Now, there are a lot of words there, so let's unpack it. Well, first, we're going to have an epithelium. And these sort of ovals are going to be a particular cell type called a keratinocyte. And the keratinocyte is the most common or most numerous epithelial cell in the epidermis. But it's a classic epithelium. So to be a classic epithelium, these epithelial cells need to be attached to a basal lamina. So this is the basal lamina. Now, I won't draw in hemidesmosomes, but recognize there will be hemidesmosomes between these epithelial cells and the underlying basal lamina. They will be attached by integrins. And it was those integrins that were defective in those individuals who have those terrible blistering diseases where the epithelium comes loose in the underlying basal lamina. But it's a stratified squamous epithelium. It's squamous because these keratinocytes, which are again are the most common epithelial cells in the epidermis, are flattened. So flattened is just, squamous is just a fancy word for flattened. And so we're going to be flattened cells. It's going to be an it's going to be an epithelium, so it sits on a basal lamina. But it's stratified, so that means it has layers. So this layer, which we're going to label the stratum basale, the basilar layer, sits right on the basal lamina. It's attached to the basal lamina by hemidesmosomes. But then immediately above that is another layer. This layer is known as the stratum spinosum. Spinosum is just a term meaning spiny. And the fact that that's the spiny layer is really an accident of history. Because when the 19th century histologists began looking at skin, what they would do is they would take a piece of skin and cut it. But in order to look at it in the microscope, they had to first fix it with chemicals, and then they had to take the water out of it and then put it in paraffin and cut it from paraffin. In the process of taking the water out, everything shrank. And when everything shrank, what they ended up in that second layer were these cells that looked a little bit like this. So they were pointed. They had these points on them. They said these are spines. They said this is the spiny layer. We now know why these things looked this way was because they continued, they continued to be attached at these sites by desmosomes. So the cadherins had continued to hold these cells together, and when they dehydrated, when they sucked the water out of these cells, cells then remained connected by the desmosomes, giving the cells a so-called spiny appearance. That's the only reason why that's the stratum spinosum. The more important idea here is that the stratum spinosum and the stratum basale, these cell layers are living cells. As you move away from the basal lamina and move from the stratum basale to the stratum spinosum, to the other layer, this is called the stratum granulosum, finally to the outside, where we now have the stratum corneum. It's called the stratum corneum because this is Greek, it refers to the horny layer, and what it basically is by the time you get out here, these keratinocytes are bags of keratin. So when you look at your skin, what you're looking at basically are dead cells on the surface. The phospholipid bilayers are mostly gone, and you're looking at a layer of keratin. But the only living cells that are in the skin are those in the stratum basale and the stratum spinosum. But the cells in the stratum basale are dividing. And so as they divide and replicate themselves, their progeny move up to the stratum spinosum, and they move up to the stratum granulosum, and then they move up to the stratum corneum. So there's this constant turnover. There's constant turnover of cells that are born on the stratum basale, but then move to the surface and slough off. We know this more or less intuitively. We know that our skin is replaced all the time. But I think one of the best examples of that is if you can remember when you were a kid and you'd go to the local Renaissance fair, you'd go someplace and you'd get a henna tattoo. Now, if you go to India, in India, it's a grown-up thing, and they have these most exquisite henna tattoos. But what henna is henna. Henna is a red-brown dye that binds to keratin. So when someone takes a brush or a stencil and brushes on that henna dye to the keratinocytes of the skin, that dye moves in and it binds to the keratin and labels the keratin a red-brown color. You can do it with hair too. We'll see a little later that hair is just a form of keratinocytes. And so it will turn hair or keratin red-brown. But you know full well that when you take one of these henna tattoos, that it'll be beautiful for the first few days, then it begins to fade, and it's gone inside a couple of weeks. And the reason it's gone inside of a couple of weeks is that these keratinocytes are continuously being replaced by the cells from the stratum basale. And so those cells that you had labeled with the henna are now dying and moving out to the outside and being sloughed off. So those are epithelial cells. They're transient. They don't live very long. They're the classic epithelium that divides and replaces itself. But we said that skin is an organ, and it's composed of two tissue types. So down here is the other tissue type. And this tissue type is said to be connective tissue proper. This connective tissue proper, in this case, is called dermis. And you'll remember from our discussion of connective tissues that what connective tissues are made of are these widely scattered cells embedded in a protein matrix they've secreted. So these things I'm trying to draw in here are called fibrocytes. Remember, the fibrocytes are the cell type that gives rise to connective tissue proper. But these cells now are embedded in this protein matrix of collagen and, to a lesser extent, elastin. If you look down at your feet and you're wearing leather shoes, what you're looking at is the dermis, probably of a cow. And what we do is when we slaughter these animals, we take the hides, we treat the hides chemically, we cross-link all of these proteins, mostly the collagen and the elastin, and that's what leather is. But what I want to emphasize is more of the living tissue. And that is the dermis is well vascularized. It contains lots of capillaries. The capillaries always have a lining of endothelial cells. One of the hallmarks, one of the characteristics of all epithelia is they themselves are not vascularized. They themselves do not have blood vessels. The blood vessels sit down away from the epithelium. And in the case of skin, all of the blood vessels, all of the capillaries are down in the dermis. This has implications for the epithelial cells because it means that oxygen must diffuse across the capillary wall out into the dermis to then feed oxygen to these epithelial cells. It also means that glucose needs to diffuse out of the blood into the dermis and out to the epithelial cells. So one of the limiting factors of these epithelial cells out in the epidermis is how far they live, how far they are away from the origins of oxygen and glucose. If you get more than a couple of millimeters away from the source of oxygen and glucose, these cells can't survive. And one of the reasons is there's just too great a diffusion distance for the oxygen and the glucose. These cells, as they move farther and farther from the stratum basale, one of the reasons they... But if you think about this for a moment and we bring it back round to the ideas we talked about just a moment ago with the henna tattoos and apply the concepts of a real tattoo, you can see the differences. You know that to get a real tattoo, that it takes a needle. You have to drive a needle down into the dermis and inject dye. If you've ever had this done, you know it's kind of painful because you're down into the dermis. You've come through some of the pain-sensing neurons that are out here at the interface between the epidermis and the dermis. You also know that you commonly bleed, and the reason you bleed is you're affecting these blood vessels. You're breaking some of these capillaries, and you're getting some blood, so you're down in the dermis. The reason that it's permanent is that there are spaces between these fibrocytes, and you can get the dye. The dye now can be deposited between the fibrocytes, and some of the dye will actually invest itself in the collagen and the elastin. So you've effectively dyed the extracellular space. Now, over time, there will be cells down here called macrophages. And the macrophages will move up into the upper layers of the dermis, and they'll begin gobbling up the dye and getting rid of the dye. And if you really want to erase one of these tattoos, you take laser beams and you basically break up the dye into very tiny droplets with the laser beam so that it has better access to the macrophages. The last minute here, let me add one cell type, and we'll come back to it tomorrow. The cell type I want to put in here is a melanocyte. And it secretes pigment. And it's this pigment that gives skin its color, and it has functional aspects of it as well. And we'll talk about that when we come back in here tomorrow.